So, Kate, I want to continue on with you as well. So I think you kind of hit the nail on the head. Right now, we are still sort of scratching the surface, right, of our understanding of learning our processes, building, establishing our processes. But at the same time, as we are trying to ground ourselves, right, and get to know this, there's also 80 other different ways we're trying to innovate, right? Whether it be to improve stability, to lyophilize these products, et cetera, right? So that adds a whole bunch of different complexities into the mix. And so, as you're looking to work with and partner with companies in this space, I often think that LNPs sort of get the short shrift in the discussion of the mRNA world, which is sad because I think that they are very, very complex beings that deserve lots and lots and lots of attention. Yes, and Ben would agree. So, where do you consistently see, as you're looking to partner and talking to companies, that you're seeing some of these complexities being overlooked, right? What are some of the biggest complexities that you think that companies need to be focusing more carefully on as they're approaching their LNP development?
It's funny, like you said, because Hopewell is on this side of market, right? So we provide a lipid, we provide formulation for different possible collaboration. And I start seeing, the deja vu, right? So, the question we asked 2 years ago and coming back again, right? So we think we solved the problem, but we don't. And I still like to think that especially for the big pharma, they have a lot of resources to different formulation and others. I still think coming back fundamentally is the lipid. It's ionizable lipid, right? So I think as we expanding our library, as we see different lipid diversity in the structure, we see that the lipid still fundamentally where you anchor your product. And if you find the right lipid with the cargo, I think formulation will be added benefit to stabilize and then translate from species to species. But if the lipid is wrong and you really tweak it with the formulation, just get it right into the first animal model, and you're falling apart in a second. So I think, I like to still think about the ionizable lipid, and that also drive what is the helper lipid, what other lipid is doing there, overall change in ionic profile of this particle. And so that's where we see the partner coming back, asking the question we thought we already dealt with, and the liver, and in the gene editing, the biotolerability, toxicity. So those are the things I think is really push us to understand fundamentally the protein corona and also the toxicity about tolerability of the lipid.
Okay. So really looking at this on a lipid by lipid level, right? Really homing in on the characteristics of especially the ionizable lipid. Okay.
You follow what I see is because there are some early success of the lipid. COVID vaccine is the two examples, and the people think one lipid will do everything, but actually it's not. And now we start coming back with very diverse cargoes. We have dealt with people with mRNA editing, all kinds of editing apparatus you can think of, and with other mRNA modifications. So then you see this particle coming together very differently, and also they behave very differently across different animal models. So that's why I actually think, we really need to look at the fundamentals of what we put it into the formulation at the beginning, and then be able to select the right lipid to go with, and then hopefully will make your development colleagues happier down the road.
Sumit, you're smiling. Do you agree? Will that make you and your development team happier?
for sure. You cannot fix these things later, right? LNP composition is so foundational, right? And Kate is so right. There's a lot that is coming in. Obviously, the pandemic vaccines would have us believe, it was great advancement, of course, but it would have us believe that it's rather simple or, more fixed composition. But there is immense innovation going out there, right? So, and
100%.
I can tell you, we internally, we have, we are still in the space of exploring which LNP compositions work the best for which purpose, right? And every time we see, once you change the LNP composition, things downstream, you cannot just translate, things from one LNP to another, right? Even with changing just the ionizable lipid, everything else you're keeping similar, things change drastically. So yeah, 100% agree with Kate on that.
Ben, you're 100%. Tell us more.
Yeah. So I think all of this conversation is great because it really is starting to touch on the fact that one size doesn't fit all when it comes to LNPs, and you really need to think about your application. So are you trying to invoke an immune response with a tiny dose? Are you trying to have a protein replacement therapy where you might be dosing a patient repeatedly, potentially for the rest of their life? So there's all these different considerations that you have to make when you're choosing the lipids that you want to use in the formulation, how they behave. So it's incredibly complex, incredibly important to try and get that right at the start if you can.
Yeah.
Which is a challenge.